guys, Sarah here. So today I'm going to be going over Punnet Squares, what they are, and how to read them and how to make them, and then also how Punnet Squares apply to corn snakes. Now, I know a lot of you who watch my videos are a little bit more advanced than this. I will be updating all of my genetics videos. The ones that I did before a few years ago, they were slightly lower quality, and a lot of that information is outdated now. So I wanted to start over, and I thought the best place to start is going to be the basics. So if you already know this stuff, I definitely understand, and check out some of my future videos for more advanced genetics things. But for this video, we're going to start with the basics, and then we're going to build on those. I know that when I first started breeding corn snakes, I was really looking for a video like this or something like this. So I'm going to make the video that I wish I had when I first started. So for those who don't know, a Punnet square is a square that calculates statistics of different things. And in this case, we're going to talk about how it calculates gene mutations or just genes on a single locus. A basic Punnet square looks like this. It's a square like that, and it has other squares in the middle. I know those are rectangles, but it is a little easier to make them kind of rectangly, just because that's how, at least in English, that's how we write. We write along that way. So if your native language is English and many other languages, they write this way horizontally. The Punnet square that has more of a rectangle going this way is just going to be a little easier to use. That's just kind of, it's not that important, but that's how I normally make mine. It's a little more rectangly than squarey. So you might kind of just look at this and think, well, I mean, it's just a square with a bunch of squares. Like, what am I supposed to do with it? Well, the best way for me to learn, or at least the best way that I learned originally, was instead of thinking of this as just like a square with other squares, think of it as more of a spreadsheet, a very tiny little spreadsheet. So for those of you who use like Excel, this is going to be a lot easier. So in Excel, you have at the top like A, B, and then C, D, E, F, G. And then on the left, you have 1, 2, 3, 4, 5, 6, 7. It goes down like that. So if you're kind of looking this as like an Excel spreadsheet, these are going to be the first 4 squares that show up on your Excel spreadsheet. And when you click on them, their labels then are like A1. And then the label, this would be B1. And then this one would be A2. And this one, B2. So if you, I'm going to make my ones kind of all look the same. But if you kind of look at this, this is actually a Punnet square. What we've just done is exactly what you would do if you were making a Punnet square when you were calculating a gene, or at least a locus. There can be multiple genes that share a locus, and we'll go over that a little bit later. I'll give you the genes that would do that, but for right now we're just going to focus on one gene on one locus. So what I'm going to do is I'm going to take this information and I'm going to kind of work it backwards. So when I say backwards, I mean what we normally have when we're doing Punnet squares is we start out with the parent information of the snakes that we have. So let's say you have a normal het anery and then an anery, just a regular anery. Then you would take that information and put it into a square like this. But we're going to go from the square that we've already made and work backwards so that you can kind of make some connections that maybe have made it difficult in the past to understand how to make a Punnet square. So we're going to go with like letters and numbers. Let's say we have a letter parent and a number parent. So our letter parent, let's just say the letter parent is going to be Dad. And the, we're going to make the number parent Mom. So number parent is going to be here, Mom, and letter parent, Dad. And the best way that I do to do this visually is, as most of you know, snakes and other living creatures have their double helix DNA. And I'm not going to go into the details of the double helix and stuff like that. That's a lot more advanced than I'm going to go into right now. But what these show is one side of the double helix came from one parent of the dad, and the other side came from the dad's other parent. And then same with mom. One side came from mom's dad, and then one side came from mom's mom. So what we're going to do is label each of these with what we have over here. So since dad is the letter parent, we're going to label one of these as side A, and we're going to label one of them as side B. And then what we're going to do with the mom is we're going to label one of them as side 1 and then one of them as side 2. So if you'll see, everywhere that there's a 2, this 2 represents this side of the DNA. Now, it doesn't really matter what genes are on which side right now. There are a couple of genes in corn snakes where it does matter which side the gene shows up on, but 99.99% of the time, that isn't something you have to worry about, and I will get into that in a future video. I'm just not going to get into it now. We're going to start with something very, very simple, and like I said, we'll build on that. So, again, here, where there is A, that represents this A, and this A. So this side is that A, and then this side is that B. And then this side is that 1, and this side is that 2. So let's go ahead and pretend that we have a pairing that we're putting together. And I know that I mentioned like an anery and a het anery, so let's go ahead and just play with that. Let's say that the dad is an anery and the mom is a het anery. So if the dad is an anery, I'm going to use N to signify the anery gene. If dad is an anery, that means, at least in this case, anery shows up on both sides. So that's not always going to be the case when you're talking about every single different gene, but I am going to start with a recessive gene like anery that doesn't have anything that's allelic to it, just the most simple gene possible to start out with, and then we will build on this in future episodes where we talk about things like tessera, or where we talk about things like palmetto, or even ultramel. Those things are going to be a little bit more complex, so we're just going to start with the most commonly occurring type of gene in corn snakes to start out with. So since anery is recessive, and basically what recessive means, it means if you have a het anery like mom, that means if anery only shows up on one side, that means that if there's nothing on the other side, anery doesn't show. And since anery can't show up with any other genes, then if there's no anery over here, that means that there's nothing over here. So I'm going to signify X, meaning no gene. We have to have a placeholder for a place where there's no gene. Otherwise, you would just have a bunch of loose ends in here that didn't have ends, not E and DS, but ends as in anery. You would have a bunch of just ends in here that don't have anything next to them, and you have to have at least a placeholder for each thing, just as a symbol for like the number 2. Because if we only put the N on one side, but we didn't put anything in for number 2, then you, like I said, you would just have N with nothing going on. You could keep the 2 and just say N and then 2, the number, just to like, you know, if that's easier for you, but I'm going to go ahead and use X to mean nothing on that side. So since mom is het anery, she has anery on one side and then nothing on the other side. And that means that she is not a visual anery. And that's going to be the case for the majority of corn snake morphs, not all of them, but most of them. Now, the dad on this side is a visual anery. So in order to get a visual anery, that means anery has to be on both, because anery is recessive. So I'm going to go ahead and say anery is on both of these and only on one of these. And to go ahead and put that into what we have over here, I'm going to go ahead and make a second Punnet square to sort of signify what we're going to be plugging in. So what we've done is we've started out with our basic Excel spreadsheet where we just have our symbols A's and B's and 1s and 2s over here. And then we've gone ahead and we've applied the A, B, and 1 and 2 to the different sides of the DNA on mom and dad. Then we take the genes that we're working on and we're putting them where the A and the B and the 1 and the 2 would be. So remember though, with anery, you can't just put another gene in here. So if you have, let's say, an anery and an amel, you can't just put amel over here because they're not on the same locus. They're in a different place. Think of it as most genes in corn snakes don't like to play with other genes. They're not friends with other genes. They're only friends with the same gene. So you're never going to find an anery and an amel on the same locus, not unless a new gene of amel pops up somewhere, which so far it hasn't, so we're not going to talk about that. But remember, this is only for one locus, which in most cases means you're only working with one gene. Now, like I said, at the end of the video, I'll go ahead and give you a list of which genes there are multiple genes of, and then we will use that in the next video to calculate the multiple genes that do play well together. But for now, we're just going to stick with this. So we're going to make a second square, like so. And instead of putting A's and B's and 1's and 2's in here, let's go ahead and plug in what we have put underneath for the gene that we're working with. So since we have an N here, everywhere that there's an A, we're going to put an N instead. So let's say there's an A up here for that one, and then there's an A here, and then there's one here. So all of the A's are covered. Same with the B's. Everywhere that there's a B, there's going to be an N. So there's also an N here, here, and here. And over here, everywhere that there's a 1, we're also going to put an N. So there's a 1 there, there, and there. And everywhere that there's a 2, we're going to put an X. And remember, X just signifies that the anery gene is not at that spot. So I'm going to go ahead and put an X there, there, and there. And now what we've done is we have made a perfect Punnet square for the gene that we're calculating. So if you have been following along with me with a pen and paper and you've been doing this, then you have made an actual Punnet square with the gene that we'd be working with. Now, I don't do Punnet squares exactly how everyone else does them. You might see a lot of Punnet squares where instead of an X, you would see like a little n instead. I find this to be actually more confusing because the capital N would mean the gene is there and then the little n would mean that it's not there. I just prefer to use X personally because it doesn't really matter what symbol you put there. All that matters is that you understand what that symbol is. So if you've seen Punnet squares or if you've used Punnet squares before where they use the capital letter and then the small letter or maybe 2 small letters or whatever to signify what they're doing, and that's easier for you, feel free to go ahead and use the small letter. But like I said, it's easier for me to know that X means there's nothing there. And it also keeps me from confusing things like a capital P and a small p. You pretty much make them the same, but they're just slightly different, and if you write them incorrectly in here, it's really hard to read. So, like I said, that's why I just pick an X, because it's easier for me to understand. So, now that you kind of have an understanding of what a Punnet square is, and how, A would go here, and then the 1 would go this way, and then you also understand how that applies to the mom and dad and their DNA strands, you've also created an official Punnet square for a real-life potential corn snake breeding. So now what we're going to do is we're going to read this Punnet square. To read the Punnet square, you just simply have to look and see what you have. What we have here, we have 2 N's there and we have 2 N's there. And this is just like Dad. Dad has 2 N's. Now N plus N, that means homozygous anery. Now the dad is a homozygous anery. Now for those of you who might be intimidated by the word homozygous, it simply means that a gene is on both sides of the DNA. Heterozygous, which is het is short for heterozygous, that's what mom is. The gene is only on one side of the DNA, not both. So heterozygous anery means it has the gene on one side and not both sides. Homozygous means it's on both sides. A lot of people think that all hets are hidden. So, like, oh, it's het anery. That means that it doesn't show the anery. Well, that's true, but it's different with other genes like tessera. Again, we will go into that in a future video, but don't immediately jump to the conclusion that because it's a het, that means you can't see it. Some genes, which are dominant or incomplete dominant or co-dominant, like I said, we'll go into all those genes in a future video, or all those terms in a future video. We're not going to go into them now, but not all genes that are hets are going to be hidden. So it's much better to say a heterozygous or a het gene and a homozygous gene or a home gene, I usually say HOM for that, than it is to say like visual or hidden, just in general. I know it's easier to sort of think about when you say visual or hidden. Just kind of keep in mind that, just kind of, you know, like I said, keep in the back of your mind that not all hets are going to be hidden. So NN equals homozygous for anery. I'm just going to say N there. And then the NX, and by the way, if you ever come across something that says XN instead of NX or anything like that, it doesn't matter what order it's in. It doesn't matter if the order is X first and then N, because all that matters is that it happens on one side. It doesn't matter which side it happens on. So the NX or XN, whichever it might happen, that's going to be het N. And so what you have here, if you want to read your results in corn snake speak, basically, is a visual anery. Half of them are visual aneries. And then our het anery, half of them are also het aneries. So this is a pretty simple one. This is probably one of the most basic you're going to come across, half and half. Now I'm going to go back in and tweak just a couple things to give you an idea of another option, another thing that might happen if, let's say, dad is also just a het anery. So anywhere that there's a B, we are actually going to make that an X. X, X, X. So now the results are a little bit different. And now we also have NX and XN, as I mentioned a little bit earlier. It doesn't matter what order these are in, at least not in this case. Like I said, there's a few cases where it does matter, but I'm going to get into that in a future video, and I'm going to say 99% of breeders are never going to have to deal with it, so it's not something I'm going to go over right now. But in this case, and in most cases, it does not matter if N came from the mom or from the dad. So we also got a new genotype, and that's what each of these are. It's called a genotype. We got a new genotype, which is XX. And XX means that there is no anery at all in that snake. So if you want to look at this and break it down, we have half het aneries that are normals, a quarter that are normals not het anery, and then a quarter that are aneries, and that's it, that are visual anery. I'm not going to go into everything that means right now, but essentially what you would say is you could go with a percentage, or what's a lot easier to say is 1/4 is visual aneries and 3/4 are not visual aneries. And not only that, but these have a 2 out of 3 chance, these normals, since there's 3 different sets of normals, 2 of those sets are het anery, 1 set is not. So there's a 2 out of 3 chance that any normal in that clutch is het for anery. Now, since you don't know which ones those are, you can't for sure pick them out because anery is recessive. Like I said, that'll be different in the future when we have different genes that we're working with. But for most genes in corn snakes, you're looking at if there's a normal that's possibly het for anery, or possibly het for any of these other recessive genes. And you get something like this where you get some normals that do have the het and some normals that don't. That's when those percentages come in. So in this case, this is where you would say the 66%. It's actually closer to 67 because if you have to kind of round up. So 3 or I'm sorry, 1 out of 3, which would be 0.333333, or about 33%, have no anery gene, and then about 66-67% do. So that's where the number 66% possibly het anery, or just the possibly het, comes from. I may go into that in a future video for other genes, but I just thought I'd mention it here in passing so that you get kind of an idea of where those percentages come from. I'll go over the 50% and other possibly hets later on. I'm not going to do that in this video. However, I did promise you that I'd give you a list of genes that do have friends that they occasionally will, play with, for lack of a better word, when they are together on a locus. So the genes that will work with other genes to make a new visual appearance are AML plus ultra. So the AML gene and the ultra gene, they can appear together. They will mix together. Let's see, Motley and Stripe. Motley and Stripe is a little bit of a weird one. We'll go over why in a future video. But Motley and Stripe can appear here and here, whereas, like I said, anery has to have a nothing next to it. Anery can only have an anery next to it or a nothing next to it. But AML, for example, AML can have an ultra next to it. Motley can have a stripe next to it. Castagna, I'm just going to say cast plus sunrise, N-R-I-S-E. Castagna and sunrise can be together. And castagna, for those who don't know, it means chestnut. The snakes have a very chestnut color. They're very beautiful, very different. And then there's also this very interesting one, hypo strawberry. I'm just going to say straw plus Christmas. I'm just going to say Xmas. I hope that I don't offend anybody by that. I put that there just to save space. But hypo strawberry Christmas, they all share an allele. And so you can have even 3 different genes going on in that, and they can all appear up in here together. We will go over that weird combination in a future video as well. I'm not going to overwhelm you with it. But these are the known different genes that can appear together in the same place. Whereas the other genes so far that we know of cannot appear with any other gene but themselves. I hope that this helped you. I hope that now you kind of have a better understanding of how to, understanding of what exactly a Punnet square is, and how, the A goes down and the B goes down, and then the 1 and the 2 go over, and then how to go ahead and transfer that information onto the DNA of the parents, and then how to, after you've renumbered the gene that you're, or relettered, I guess, the gene that you're talking about, how to plug those into a new Punnet square, and then how to read the results. I really hope that this helped you. Please let me know in the comments if there's anything that I missed or anything that's vague or anything that you'd like explained a bit in a little more detail in the future video, because I would be happy to do it. Like I said, I plan to redo all of my old genetics videos in this format so that it's a little bit easier to watch and so that you guys can kind of see it on a slightly bigger scale, and of course you'll get updated information. So thank you for watching, guys. Appreciate you. See you guys in the next video.